Alright, this is going to be my DNA sequencing video, like the whole process and what it includes, and these are my visual aids, I guess. Like in my last video, I made my markers and the little tiny board that I have. So the first step in DNA sequencing is basically going to have from PCR, you're going to have your little DNA strands, your template DNA, and you're going to have your, I don't have my glue on me. That was loud. So instead we're going to use red right there, you're going to have that one, and then we'll use green. I just got a marker in my mouth, that's not nice. So I hold my phone with one hand. That does not taste good. Alright, you got your primers there, you know what I mean? Then you're going to have your TAC polymerase go down that way, and you're going to have it go down that way, and then there's your two template strands. So with these strands, you have like 40 versions, like 100,000 versions of these, however many you need, so you can make less errors. The more you have, the less errors you make, basically. Once you have this, you basically add it in you have your polymerase and your nucleotides all set up, and then after this, you're gonna, there you go. After the where's another marker? Oh boy, I lost my one. There it is. No, it's not. We'll just use. I lost my marker. Nice, mid video. All right. So you're gonna split the. That's really uneven. You're gonna split the You basically put the your DNA sync. You put your DNA templates. You have 4 groups of them. One of them is gonna be DDNTPS. The other one's gonna be DDNAPS. DDNGPS. GPS jokes. And the last one's gonna be DDNCPS. What those stand for, if you see, is this, that one, the A, T, C, and G are the only thing that change. Basically, that stands for thymine, adenine, cytosine, and guanine. Basically, what this will do is make it to where you can have, like, we'll do G is gonna, we'll do G is red, and C is green. I think that's gonna work. No, we'll screw that. We'll do not C, we'll do T. So we're gonna go T, 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 and then we're gonna get a G. So if we have that, if it's in this group, then it's gonna cut off right there, and we're gonna have our strand right here. And with that, and then in the next one, let's say if we're in T, we have guanine, 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 and then at the very end we have one thymine that's going to cut off right there, and once again we have another strand. Now once we have this, you're basically going to throw it in gel electrophoresis, and it'll be, in the fourth step, the last part, you put these DNA samples in the gel electrophoresis, which has an electromagnetic pulse in it, which is going to affect the DNA to move in it. So the longer the strand, the more it's going to move, or the more it's going to move forward. So you'll be able to read it with the naked eye. And another thing, these all the DDs, all those little primers, I believe they're called, are gonna basically make it fluorescent so you can see the nucleotides better. and it's just going to look like this with random strands going down, with longer ones being at the end, and then like little middle ones in the middle, and then at the very back you'll have tiny little ones because they're tiny, and then you have to, like, the more you go, the big, you get the point. That's gonna be my video on DNA sequencing. These are the citations I have. Okay, paper, stop doing that. And like those are my little PowerPoint notes or whatever, or notes, not the PowerPoint.